Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. Today we are going to talk on the module which will describe or discuss lipid bilayers, micelles and liposomes from the paper which is Structure and Function of Biomolecules 2. Fatty acid anions, because under physiological pH the carboxyl group of the fatty acids, as we pointed out, actually gets ionized. So what is present are not fatty acids but their anions, so that's physiologically relevant. So fatty acid anions, bile salts which are basically steroid in nature, very necessary during fat digestion, and phospholipids. So these are three very different kind of compounds in terms of their structure. Their structure is very different. Bile salts don't have a structure anywhere remotely similar to the simple fatty acid anion. So what is the common feature among all these molecules? The commonality is that they are all classified as lipids because they are soluble in organic solvents. That's the definition of lipids with which we started, and that's the way it is. But why are they all soluble in organic solvents? That is because all of these have a structural component, and that's the larger component of the molecule, which is non-polar in nature. It is also important not to forget about the small polar structural component. The result of having this large non-polar component and also a small polar structural components actually makes all these classes of molecules as amphiphiles. In the case of fatty acid anion, the ionized carboxylate thing is a small polar head. The long hydrocarbon chain of the fatty acid is the large hydrophobic or non-polar structural component. So fatty acid anion is an amphiphile. In the bile salts, the condensed ring system of the steroids is the non-polar part, but then you have those small substituent groups which are charged, which are polar. So bile salts are also amphiphile. In the case of phospholipids, those polar phosphate, linking through the phosphate moiety and sometimes the polar bases which are attached, they constitute the small polar structural component. The rest of the molecule, including the 3 carbon of the glycerol, because please remember that in most of these cases the glycerol chain, the 3 hydroxyl groups are not free. Even the upper 2 are actually linked generally by, for example, fatty acyl chains, which are long chain, again long chain non-polar in nature. So phospholipids are also amphiphiles. So such molecules are also called surfactants in chemistry, and many a times we call them detergents. So that can at times be confusing, because in chemistry the word detergent is used as synonymous with a surfactant, but you are more familiar from in your daily life as a detergent. It's something which cleans, and that arises because those detergents actually, their cleaning action is based upon the surfactant nature of these molecules. So it's better to be very clear that in what context we are using what words. So the objective of this module are to understand the mechanisms of aggregation of molecules to form micelles, bilayers, also to learn about how lipids can form monolayers, bilayers, soap bubbles, and vesicles. We'll also learn briefly about these important entities called liposomes, how one can prepare them, how one can purify them, and what are their structures like. So the basic concept involved is that many lipids have this small polar push component and a large, normally a larger part is a non-polar part, and these lipids act as amphiphiles. And how these amphiphiles, depending upon variety of parameters, and bulk of these parameters belong to the structure of their larger non-polar component and smaller polar components, they aggregate, and they can aggregate to form variety of aggregated structures. Some form bilayers, some form micelles, and some form liposomes. With this module, we start our discussion on property of many lipid molecules to associate to form supramolecular assemblies. These associated structures can be of various shapes and dimensions. The aim of this discussion is to lead to details and formation and function of biomembranes. That is not to say that the micellar structures and liposomes themselves have no relevance. Liposomes in fact have emerged as a part of a drug delivery approach. In a retrospective appraisal, these are viewed as a nano drug delivery systems. Micelles or microemulsions are also involved in physiological processes such as digestion of lipids like fats and oils. Micelles are aggregation colloids. This class of colloids are formed when species aggregate to reach the colloidal size. Lipids by no means are the only class of compounds which form micelles. Micelles formation requires amphiphilic nature, however. So this kind of a structure has a polar head and a hydrophilic tail, or more polar heads or more hydrophilic tails in whatever ratios. So both small molecular weight compounds as well as larger molecules can form micelles. Amphiphiles or surfactants can be ionic surfactants or non-ionic surfactants. Ionic surfactants again can be anionic or cationic. Almost all biochemists are familiar with sodium dodecyl sulfate, which is used in SDS page electrophoresis. That's an anionic surfactant. Citab is a common detergent or surfactant which is cationic. Bridge, another surfactant which is widely used in life sciences, is common example of a non-ionic surfactant. Or nicotinamicides, as these are called by the people who work with surfactants. There is a class of polymers called pluronics. They are an important example of non-ionic surfactants of larger sizes. Milk contains casein molecules which are present as micelles. Casein molecules form micelles with quite intricate structures. Casein thus is an example of a protein forming the aggregation colloids or micelles typical of a surfactant molecule. Phospholipids can also be termed deuterionic surfactants. The well-known phospholipid lecithin or phosphatidylcholine has two hydrophilic tails made up of acyl group of long chain fatty acids. The negative charge on the phosphate and the positive charge of the choline constitute deuterionic polar head. In dilute solutions of surfactants, the molecules exist as isolated species. As the surfactant volume fraction increases, micelles are formed. Increasing this further gives rise to lyotropic metaphase, which are called liquid crystals or liquid crystalline phase. These phase transitions are shown in this slide. The self-assembled structures are important in many areas. In biology, micellization is relevant in areas such as digestion of fats and oils, vesicles and their reversible formation is also important in many biological processes. Biomembranes are in fact a mosaic of bilayers. Surfactants are used in cleaning detergents, textile industries, in cosmetics, in industries related to paper, paints, food, metallurgical process such as froth flotation, which you would have studied in your school chemistry, you may recall, also involves the use of a surfactant. Micelles as well as reverse micelles have both been used to carry out chemical reactions, biotransformations, and have been described as nanoreactors. Reverse micelles are in fact another well-known example of a low water reaction medium for enzyme catalysis. As we can see, micelles can differ in both shapes and of course sizes. We now understand that few parameters are definitely important in determining the shapes and sizes of these micelles. Head group size, that is the so-called polar head part of the molecule, has an important influence. Ionic strength of the medium is very important. While in vitro this can be controlled, in biological systems physiological conditions of course define it. Not only polar head, the hydrophobic tail of course also is an important parameter which determines the shape and the sizes. The common description of micelles is in the many books is actually an oversimplified picture and actually misrepresents the reality. The first thing to be understood is that the core is not dense. This is because tails are not straight arrays as is normally shown, but they are actually entangled as shown in the lower part of this slide. The heads are not so neatly aligned. Not all micelles are shape persistent. Both micelles and reverse micelles are dynamic structures, and there are definitely exchange of materials through micelles. The percentage amount of water varies from micelles to micelles. Pluronics, the polymers which form water core, for example, which is about 30% on a weight basis. There are various techniques which allow us to directly look at sizes and shapes of the micelles. Cryotemp, for example, is one such technique, and this involves rapid cooling of the thin layer of sample in liquid ethane. Small angle X-ray or neutron scattering are another two important techniques. An important parameter related to micelle formation is critical micelle concentration, or CMC as it is normally called. Critical micelle concentration is the concentration above which micelle formation is observed. Below this value, that is the CMC, the surfactants are adsorbed at the layer. We have already known or encountered this phenomenon while discussing lipase biocatalysis. The natural substrate of lipase form micellar phase beyond CMC. This led to opening up of the active site molecular lid and hyperactivation of lipases in the case of several lipases. Surface tension values at CMC show abrupt changes. CMC of ionic surfactants is generally higher as compared to the non-ionic surfactants, which implies that among lipids and phospholipids, non-ionic ones will form micelles at earlier molar concentrations. Equivalence conductivity also changes abruptly at CMC in solutions of the surfactants. Hence, measurement of surface tension and conductivity provide useful ways to monitor micelle formation. In general, longer tails of the surfactants decreases CMC of the surfactants. This makes sense as the hydrophobic interaction among tails provide the main stimulus for micelle formation. Longer the tail, more is the hydrophobic interaction. Some other useful parameters about micelles can be identified. Size and CMC are two which we have already talked about. CMC of common ionic surfactants has been found to be in the range of 10 raised to the power minus 3 or 10 raised to the power minus 2 molar. As already indicated, CMC of non-ionic surfactants is lower. CMCs of common non-ionic surfactant is in the range of 10 raised to the power minus 4 to 10 raised to the power minus 3 molar. The polar heads of non-ionic surfactants will not have any electrostatic repulsion due to some, and hence self-assembly will be easier. Aggregation number refer to the number of molecules which self-assemble to form a single micelle. This number is very different from micelles made from ionic and non-ionic surfactants. The aggregation number for non-ionic surfactant is in the range of 10 to 170. For non-ionic surfactants, the corresponding range is much larger and is from 30 to 10,000. The solubility of surfactants depend upon the temperature. The solubility is less at low temperature. This is followed by a narrow range of temperature in which the solubility rises rapidly. For all systems, there is a temperature beyond which no micelles are formed. To repeat, micelles are colloids. If the solubility is high, substances form true solutions and not colloidal suspensions. The solubility versus temperature curve and CMC versus temperature curve intersects at a particular temperature. This is shown in this slide. This temperature at which these two curves intersect is called the Kraft point, and the temperature is called Kraft temperature. The Kraft temperature has a significance as this can be visualized as a melting point of the self-assembly. In food processing industries, Kraft temperature is used as a practical parameter to design processed foods. Various models have been proposed over the years to explain micelle formation. The first was stepwise growth model. This viewed association as a continuous process. This would predict species of various sizes present at any time. It would also rule out abrupt changes at CMC. On the other hand, a model called the closed aggregation model views micelle formation as a cooperative phenomenon. Later on, some other models were suggested which are based upon defining some new parameters for the micelles. So this slide shows the, the discussion on the packing parameters. It's the Ishrakleshevi in 1976 who introduced this concept of the packing parameter P, which was defined as shown here, and the various symbols are as explained there. So what do we see here is that for common surfactants which have a single tail, the relationship is like this. So this is the whole discussion is revolves around the concept of the packing parameters, and the slide, the left hand side illustrates that what is the physical significance of this. Yeah. What are the predictions of the packing parameter model? Big polar heads will lead to a large AE, and micelle shape will approach spherical shapes. On the other hand, small polar heads will result in an associations which form lamellae or bilayer formations. Charlie Tenfold, who contributed so much to the importance of hydrophobicity in protein structure, gave another model which is called free energy model or phase separation model. According to this model, micelles are microphases. Considering the chemical potential, the equation concerning the two is shown in this slide. So the transfer term will result from minimizing the contact between tail and water. Interface term arises out of the residual contact between non-polar tail and water. The last term represents a repulsion between polar heads. To sum up, the model stated that the tail transfer decides the aggregation. Residual contact parameter promotes the association, and head repulsion restricts the association. Both packing model and free energy model are able to predict some behavior of surfactant systems correctly. For example, packing model correctly predicted that in case of ionic surfactants, addition of salts will increase V0 by AeL0 param number. Consequently, it predicted correctly that spherical micelles will become cylindrical micelles. Packing model also predicts that for same equilibrium area AE, the ratio of V0 to AeL0 will be 2 times for a surfactant with double tails. This results in formation of bilayers instead of spherical or globular micelles. This gives us a clue as to why nature has chosen lipids such as phospholipids with 2 tails to be part of the biomembranes. Biomembranes, as we will see, involve formation of bilayer by these lipids. The packing model also predicted correctly that increasing temperature will decrease the steric repulsion between heads in case of PEO micelles. Such micelles change from spherical to cylindrical shapes. The tail, according to the packing model, affects the packing parameter and hence influences the aggregation. The hydrocarbons pack differently in bulk as compared to when they are part of the tails of the surfactants in a micelle. Detailed analysis based upon simulation experiments show some interesting and non-obvious conclusion. One is that the tail length affects the head group area and it thus influences the shape. So, according to thermodynamics, for at least low molecular weight surfactants, delta H is about 1 to 2 kilojoules per mole. Hence, enthalpy does not favor micellization. Delta S is about 140 joules per kilo. Thus, entropic factors favor micelles formation. It is actually the properties of water which facilitates micelle formation, and these properties are high surface tension, high cohesion between water molecules, water has high dielectric constant and high boiling point. The non-polar species placed in water results in water molecules forming clathrate structures around them as first shell. These more open iceberg structures involve ordering of hydrogen bonds among water molecules with a large entropic cost. When the temperature is raised, hydrogen bonds are broken with increase in entropy but at the cost of enthalpy. A small size of water molecule makes it difficult to find a cavity for the nonpolar solute. Thus, the insertion probability for the solute is higher than a simple molecule like normal hexane for small cavity sizes. For 1 angstrom cavity radius, it is much less and nearly equal for both liquids. Lipids, with their solubility in organic solvents, all have nonpolar components. When polar and charged structural motifs become a part to convert them into amphiphiles, interesting associations take place. These associations ultimately have the basis in the hydrophobic effect. With this background, we are in a position to appreciate the behavior of lipids to form bilayers, liposomes, and fluid mosaics. The choice of polar heads and tails, as we have learned, dictate the nature and results of self-assembly. So, lipid amphiphiles like glycerophospholipids, sphingolipids, and even soap can all form the aggregates in the form of monolayers, micelles, and bilayers. Soap, as you know, is the sodium or potassium salt of a long chain fatty acid. Soap molecules gently spread on water surface, or more properly called the air-water interface, and then they form monolayers. The charged part naturally interacts with water, and non-polar tails are oriented up on the air side. When soap molecules are immersed in water, micelles are formed. As always, polar heads are oriented towards outside the bulk medium of water, and tails are inside, as we have discussed in the general picture of micelles. Soap bubbles, on the other hand, are bilayers of fatty acid anions forming a spherical shape which enclose a thin water layer. The formation here consists of the tails stacked together. Polar heads form the middle of this sandwich, that is where the thin layer of water is there. The interior of the bubble and the outside is same as the outside of the soap bubble, that is air. Tails of each layer extend into air on each sides, hence in this structure. Air and its component gases are not highly soluble in water. Relatively, air is non-polar as compared to water. Hence, thermodynamic stability favors the orientation of soap molecules as is observed in the air bubble. The main components of the biomembranes, glycerol and phospholipids and sphingolipids, can also form monolayers under controlled conditions. However, left to themselves, they spontaneously form bilayers. Unlike soap with one hydrocarbon tail, these have two long chain hydrocarbon tail. So that is how a typical lipid bilayer looks like. The two chains of the complex lipids are incapable of packing well in a micellar structure. They instead form bilayers. The choice between micellar structure and bilayer structure is decided by packing parameter considerations, and that is why we discussed that parameter earlier. Unlike soap bubble bilayers, the bilayers of these complex phospholipids have aqueous phase on both sides of the bilayer leaflet. Hence, polar heads are oriented towards each surface. The tails form tail to tail orientation. Not all lipids can form bilayers. It is found that the cross-sectional areas of the tail and the head have to be nearly equal to facilitate the bilayer formation. It so happens that this is so for glycerophospholipids and sphingolipids. Lysophospholipid have larger head and a single acyl chain. Similarly, in cholesterol and other steroids, the non-polar part is too large. Neither class of lipids form bilayers. Bile salts, for example, you will recall, form micelles during digestion. Liposomes are another interesting structures. These are formed from phospholipid bilayers joining ends and enclosing an aqueous compartment. The aqueous compartment and liposomes allow delivery of water-soluble molecules like drugs. Liposomes of similar size range and shapes can also be formed by dropping the lipid into water through a fine needle. Liposomes are fairly stable structure and thus can be separated or purified by centrifugation, gel filtration and dialysis. Typical of the lipid aggregates, these also show. So in summary, in this module we learnt about mechanism of micelle formation, bilayers and liposomes. In bilayer formation, increase in solvent entropy exceeds the decrease in phospholipid entropy to drive the process. A phospholipid in bulk water has ordered array of water molecules around the hydrophobic tail. Bilayer formation abolishes this order. Another reason liposomes have been a good choice for drug delivery is that they can fuse with the membranes of the target tissues. That also is due to the property of cell assembly of these interesting amphiphiles. Thank you.